Today we are going to talk about the cell history. Now we are going to give an introduction about what is the cell history. When we say a cell history, we must know what's meant by a cell. The cell comes from a Latin word called cella. Its meaning is small room, the basic structural, functional, and biological unit of all known organisms. A cell is the smallest unit of life. Cells are often called the building blocks of life. Cells is discovered after microscope is invented. Without microscope, we wouldn't have known about the cells. In 1665, Robert Hooke first observed cork. The cork is a dead tree bark. He refined the design of the compound microscope and published a book. Then Robert first observed cork. He saw boxes that reminded him of tiny rooms. He named the empty boxes cell. And we are having two pictures to describe what I'm saying now, the dead cork or the dead tree bark. And now we are going to talk about how the cell theory is concluded by Max Schleiden. He is a German botanist. He concluded that the structure of planets were composed of cells. He extended Schleiden's view and concluded that all life is composed of cells. Then Schoen, the German physiologist, he extended Schleiden's views and concluded that all life is composed of cells. Now we are going to talk about the cell theory. The cell theory is divided into three parts. First, all organisms are made of cells. Second, the cell is most basic unit of life. The third part of cell theory is concluded by Rudolph, who concluded that the cells arise from pre-existing cells. And now, what is the big problem with the theory? The scientist is not understanding how life was begun. The shortcomings and drawbacks of cell theory are viruses are considered as acellular entities or organisms that do not have cell machinery, yet they are taken into account as organisms in the cell theory. Schleiden and Schoen didn't know that the mechanism of the cell. And now we are going to talk about the cell diversity. What is meant by cell diversity? The shape of the cell depends on its functions. In multicellular organisms, different cell types tend to be organized into distinct groups or tissues according to their function. The most complex organisms, like humans have evolved highly organized arrangements of different types of cells. We are going to have two examples of diversity. The first one, the nerve cell, second, red blood cell. But now we are going to talk about the nerve cell, its shape, its function, and its advantage. Its shape, elongated and branching. Its function, it sends messages to and from brain. Its advantage, signals can travel a variety of direction, and we have a photo for the nerve cell. Then we are going to take the red blood cell. The shape is this disc-like shape, and the function, carry O2. Advantage, roll through narrow veins, areators. And we are going to have a picture for red blood cells. And now we are going to have the cell categories. It consists of two types, the prokaryotic and eukaryotic. The prokaryotic, they are the cells that are without a nucleus or other membrane-bound organelles. We are going to have examples for prokaryotic, such as bacteria, first cells life on Earth, with the fossils 3.5. And we are going to have a photo or structure for prokaryotic. As we see, eukaryotic, cell with a nucleus, membrane-bound organelles, more complex cells, evolved from prokaryotics. And now we are going to have four pictures, means four examples for eukaryotic. Type of Protistia, mushroom type of fungi, tree type of plant, frog type of an animal. And now the last thing, the eukaryotic evolution. It comes from the endosymbiose theory. Chloroplast and mitochondria were free living organisms. The evidence for this, that mitochondria and the chloroplast contain their own DNA, They replicate and contain their own ribosomes. The process, they have larger cells engulfed chloroplast and mitochondria. Larger cell has an advantage. Each